As the name indicates, most ciliates feature large numbers of cilia, tiny beating hairs. These heterotrophic unicellular protists are 10 to 3,000 micrometers long. Their cilia are usually arranged either in longitudinal rows or in spirals around the cell. Are anchored to microtubules beneath the plasma membrane, see Chapter 5, and they beat in a coordinated fashion. In some groups, the cilia have specialized functions, becoming fused into sheets, spikes, and rods that may then function as mouths, paddles, teeth, or feet. The ciliates have a pellicle, a tough but flexible outer covering that enables them to squeeze through or move around obstacles. All known ciliates have two different types of nuclei within their cells: a small micronucleus and a larger macronucleus. Figure 29.16. Macronuclei divide by mitosis and are essential for the physiological function of the well-known ciliate paramecium. The micronucleus of some individuals of Tetrahymena piriformis, a common laboratory species, was experimentally removed in the 1930s, and their descendants continue to reproduce asexually to this day. Paramecium, however, is not immortal. The cells divide asexually for about 700 generations and then die if sexual reproduction has not occurred. The micronucleus in ciliates is evidently needed only for sexual reproduction. Vacuoles, ciliates, form vacuoles for ingesting food and regulating water balance. Food first enters the gullet, which in paramecium is lined with cilia fused into a membrane. See Figure 29.16. From the gullet, the food passes into food vacuoles, where enzymes and hydrochloric acid aid in its digestion. Afterward, the vacuole empties its waste contents through a special pore in the pellicle called the cytoproct, which is essentially an exocytotic vesicle that appears periodically when solid particles are ready to be expelled. The contractile vacuoles, which regulate water balance, periodically expand and contract as they empty their contents to the outside of the organism. Pairing of two paramecia. Two paramecium individuals of different mating types come into contact and join at their oral surfaces. 2. Meiosis of micronucleus. In each paramecium, the diploid 2N micronucleus undergoes meiosis, producing 4 haploid N micronuclei. 3. Degeneration and division. Out of the 4 haploid micronuclei, 3 disintegrate. The remaining 1 divides by mitosis, forming 2 haploid micronuclei in each cell. 4. Exchange of micronuclei. The 2 paramecia exchange 1 haploid micronucleus with each other through a cytoplasmic bridge. This is the actual conjugation process. 5. Fusion of micronuclei. Inside each paramecium, the newly received micronucleus fuses with its own remaining micronucleus, forming a diploid micronucleus, 2N. Restoration of nuclei. The macronucleus disintegrates, and the new diploid micronucleus divides by mitosis to produce 2 identical diploid micronuclei. 7. Formation of new macronucleus. Of these 2 micronuclei, one becomes the new micronucleus for the cell, and the other develops into a new macronucleus. Summary. Through conjugation, paramecium exchanges genetic material without forming offspring immediately. This process increases genetic variation and rejuvenates the cells, allowing them to continue reproducing asexually afterward.